everyone. I'm Lisa Miner. I work with Multispan. Today, we're going to be talking about GPCR assay bias or receptor bias and how that can affect your assays and what you want to look for in drug discovery. I work with Multispan. Multispan is a company in California, and our main focus is providing cell lines and assays for people in discovery research. And our main area of focus is GPCRs. So today, I'd like to introduce Terry Kanakin, who is my partner in this presentation. So Terry, you want to introduce yourself, please?
Hi, folks. Hi, I'm Terry Kanakin, and I've spent my career studying GPCRs, allosteric receptor function, most of it in industry, Burroughs Wellcome, GSK, but I'm currently at UNC School of Medicine. Previous to about 10 to 15 years ago, receptors were thought to be switches. And so agonists of varying efficacy were thought to impart a uniform signal to the cell of varying strength. As we got different assays to start measuring what that signal was instead of just cell effect, we could now dissect it into GS, GI, GQ, beta-arrestin, and we realized that the cell was getting a mixture of signals and that different ligands gave different mixtures. In other words, efficacy had quality as well as quantity. And so bias is where a ligand will direct the receptor towards a certain signaling pathway at the expense of others. So now you can actually direct where this signal goes. If we can control this, this is a tremendously powerful tool for therapy because we could de-emphasize harmful signals to a cell and emphasize good ones. Moreover, you might ask, well, why do I care about bias? Because you can't assume a synthetic agonist is going to give the same quality of efficacy as the natural hormone or the neurotransmitter. You're going to have to quantify that and see if there's better signals or your compound could actually be giving the wrong signals. So bias and the ability to quantify it, detect it, is an extremely important tool now, and it has basically revitalized the receptor field.
So why don't we go to the first slide and you can give a description a little bit about what agonist signaling bias is.
Sure. This is an example from our HIV infection program years ago. One way to prevent HIV infection is to internalize the receptor that mediates binding of HIV to the cell. And that receptor is the chemokine receptor CCR5. As ligands interact with this receptor, they can internalize it. And if it's internalized, this prevents HIV infection. But not all ligands have an equal power to do this. This is bias now. Some are very good at it and some are not. As you'll see in the curve to the left, we have the curves for IP1, which is a physiological signal, and internalization. And the chemokine CCL3L1 is pretty well equiactive. It's a very good internalizer. Most chemokines are not, and that's shown on the right. The chemokine CCR3 shows that the internalizing curve is shifted far to the right. It's a much poorer internalizer. This was an extremely useful assay because we used it to guide chemists to give synthetic chemokines that would be good at internalizing. So here's an example of a physiological usefulness of bias in terms of taking two signals and emphasizing the good one.
What about with low and high tissue sensitivity?
That's very important. In order to quantify this and to detect it and so forth, you must have two curves for both pathways, a curve for each pathway. On the slide, you see on the curve to the left, you see one signaling pathway, and then the compound did not produce any effect on the other. Now, you can interpret this one of two ways. You could say, oh, my compound is perfectly biased. It does not activate the second pathway at all. Or you could interpret it and say, oh, my assay is not sensitive enough to detect that second pathway. This is a very important difference because you will give the compound in vivo and it will encounter tissues of varying sensitivity. And if it encounters a more sensitive tissue, you might actually unveil that second pathway. And that's shown on the curves to the right. In a more sensitive tissue, you might actually see that curve. So it's critical to have an assay that can give you those curves. Routinely, G protein signals are highly coupled, sensitive, whereas beta-arrestins are not. And so you'll frequently see compounds that will activate G proteins and not beta-arrestin. It's important to have an assay to see if that's really true or the assay is just not good enough. On a receptor level to control sensitivity, the next slide shows what can happen. This is the muscarinic receptor and responses to carbachol and oxytryptamine. In a poorly coupled tissue, you will see a curve to carbachol, no curve at all to oxytryptamine. So you can interpret that as oxytryptamine's an antagonist, and you'll never see a response to it. However, in a sensitive tissue, you'd see that you were wrong. You see that oxytryptamine produces a powerful response. So just having the assay to detect that potential option is really, really important.
One of the ways that we can increase the sensitivity is to increase the amount of material within the cell that's involved in signaling. And part of that material is arrestin. And so we at Multispan have developed a new assay which can measure arrestin signaling, but it also can be used to look at to increase the amount of arrestin in a cell. And you can actually monitor how much arrestin is in the cell by its signal. So let's go to slide 4, where you can see what our technology is. And what this basically is our arrestin technology, where we have a nanoluciferase that we have put part of an arrestin on, and then a cell membrane targeting sequence that has the other part of the arrestin. And so when the GPCR, which is not tagged with anything, is activated, the arrestin can move to the surface, meet its partner for the nanoluciferase that's in the membrane, bind, and then you have a light signal of light. The benefits of this essentially is for the assay itself is you can measure arrestin, and you can measure arrestin in a cell where you can measure your GPCR activity as well. And since the GPCR is not modified, you have a better sense of that your signal is due to the arrestin and not due to anything you've done to the GPCR. But how can you use this to increase the amount of arrestin in a cell? Well, on the next slide, slide 5, you can see when you add additional amounts of arrestin, we get increased amount of arrestin signaling in that cell as measured with our technology. So we do get an increase in background, but that means that we have more arrestin in that cell. And so you'll be able to measure whether there is an effect on arrestin signaling that's because, you know, you can change the amount of arrestin. So you can kind of dial it in to see whether you can dial in a signal or whether you still have no signal to arrestin at all. So that's one thing that this particular technology will allow us to do. So that's all I want to say about that. But one thing I wanted to ask you, do you have anything in addition to add to what we're talking about? But after you do that, I want to ask, are there any compounds now that are approved for clinical use that are biased, either designed to be biased or were accidentally found to be biased, or, you know, so because that's the question I do get is what's it matter whether it's biased or not? Does it mean anything?
Yeah, it's actually a question I've heard too. Well, just to sum up, to emphasize that bias is readily quantified in vitro, but you have to have the curves. It's not good enough to say, I don't have a curve to this compound, it must be inactive. So being able to control the sensitivity of the assay really rules. If you don't have that, then you really can't quantify bias. With respect to the physiological relevance of bias, first of all, anything new is thought to be rare when it's first discovered. And so 15 years ago, it was thought to be an odd effect found for some compounds. There's not a receptor I have seen reported that does not demonstrate it. Why is that? Because it's natural allosteric probe dependence. It's nothing special. It's the way proteins behave when they change their shape. That reflection with different interactants is not constant. So you will always have bias. So I would stress that when you have a synthetic compound, you should actually assume you will get bias. You may not, but you certainly shouldn't treat it as if it's an odd event, because it isn't. In terms of the history, are there any approved? There have been a few designed molecules that were tested. Probably the best one was the angiotensin molecule for heart failure. And it was active, but it was not any better than an unbiased compound. And so to date, there's no approved drug that was designed to be biased. However, there are drugs that are currently being tested, for example, opioids that have less ability to cause respiratory depression that are doing well. Retrospectively, there are compounds people have looked at, good compounds, and said, okay, why are they good? And they were found to be biased. Now, whether they're good because they're biased or they just happen to be biased, we don't know. But there are compounds, carvedilol and so forth. And so it's sort of a phenomenon that's with us. We can't ignore it. It'll be present in all of our programs, and it behooves us to understand it and quantify it, measure it for our new molecules.
Okay, I agree with you. I think it's a really important thing. I think it's very important to understand your biology of your molecules. And I think the missing piece is to understand what your bias is in the tissues that you're interested in, and how disease changes that bias or change, so that we actually, when we're doing testing, we can figure out exactly what we're really looking for and what is the pattern of activity that is the most important. That seems to be something that we don't have that translation yet, I don't think.
Actually, that is a good point. I'm happy you raised that because quantifying bias really is looking for the exemplar, the molecule that stands out. So you have a program and you screen and you quantify activity, and then you quantify bias and you take your extremes. You may not know what it means, but you push those forward and look to see what they do in more
Yeah.
complex systems to get phenotypes. The field right now is the difficulty in translating the in vitro bias you quantified, because there are other things that can intervene in vivo. So that's probably why it's going to be tricky to translate that. But the first step is to detect it in your compounds, and in vitro assays are essential for that.
Yes, and they're easy to do.
Yeah.
So, so thank you very much, Terry. I really enjoyed our conversation. And for those who are interested, please visit multispaninc.com to connect with Terry or me, or to find out more information from Multispan. And thank you very much.